Right. Good day. We are back at the part 3 of the staining. Last stream was interrupted, and so we were at endospore staining, the staining specific structures. Endospore staining is like staining the endospore, coloring the endospore to differentiate them, from the bacteria. Like even if the bacteria is not present in the environment, if the endospore is present, that means the harmful characteristic of the bacteria still remains. And staining the endospore is a tough job, or you stain the endospore by rough methods because endospore is usually resistant to external environments, and it is formed by certain bacteria like, for example, Bacillus and Clostridium. And we were here at the shapes, that the endospore that is produced by bacteria has different shapes, like it can be spherical, elliptical, can be smaller, and you can say larger than the parent. It doesn't necessarily have to be the same shape or size as the parent bacteria. Endospores are not stained well by most dyes, but once stained they strongly resist decolorization. First they resist colorization, but when they are stained they resist decolorization. So this property is the basis of the most endospore staining methods. So figure 2.4 or 2.14e is here. E. So this is it. Look at this. Vegetative cell means these are the normal cells that lives and grow and die. Well, the red ones are the endospores, right? And it is done by staining, of course. And in the Schiffer-Fulton procedure, we already discussed two procedures. This is the third one. And then this procedure is for endospores. So endospores are first stained by heating the bacteria with the malachite, M-A-L-A. How do you cover malachite green? What kind of green is that? Wait. Malachite green. There's nothing special about it, right? And wait, why is my Google window not working? Right, here it is. So they are saying we first heat the bacteria with this stain, which is very strong stain that can penetrate endospore, and by heating, it weakens maybe the endospore a little bit. It's not like heating can do damage to endospore in the first place, but it might help with the dye to enter it. After malachite, I don't know, malachite, green treatment, the rest of the cell is washed free of dye with water and is counterstained with safranin. They are saying that they show resistance to decolorization after dye, but still they wash it with water and counterstain with safranin. This technique yields a green endospore resting in a pink to red cell. Green endospore resting in a pink to red cell. Okay, so the endospore is inside the cell, right? Inside the cell. So when you put that malachite green, it penetrates even inside to the endospore. So, and endospore is resistant to washing, so it will never retain another color other than the first stain that you stained them on. But the cell, on the other hand, is different story. It can be washed and another stain can go above the previous one. So the endospore is of green color while the cell itself is the pink color. In this morphological microscopical picture, they didn't show the green color of the endospore. They are just showing the red, pink cell, general cell that is producing the endospores. Now the flagella staining. We did the capsule staining, we did the endospore staining, and now we are going towards the flagella staining. Provides taxonomically valuable information about the presence and distribution pattern of the flagella or prokaryotic cells. So flagella just describe the valuable information about taxonomy of the prokaryotic cell. Flagella is an important characteristic, just like the cell wall and capsules in the case of prokaryotic cells. From just the flagella, we can, differentiate between bacteria to a certain degree. Prokaryotic flagella are fine, thread-like organelles of locomotion that are so slender, about 10 to 30 nanometer in diameter. They can only be seen directly using electron microscope. So they are that small that they can't be visible to the compound microscope. So if you do study a sample under a compound microscope and has flagella, it means that you will be completely losing the identification of the bacteria, because you will be wrong about it, because you won't be able to see flagella. Flagellar stain of the proteus. A basic stain was used to build up flagella, and this is how the flagella looks like. When the prokaryotic cells have many types of flagella, many numbers of flagella. It can be on both sides, it can be on one side, it can be covering the whole cell. So to observe them with a light microscope, the thickness of flagella is increased by coating them with mordants like tannic acids and potassium alum, and then the staining with the paracyanil paracyanilline. What a word! Paracyanilline. Leifson method or basic Fuchsin gray method. So they are saying if you want to see them under the light microscope or even compound microscope, you have to stain them, and before staining them you have to use the mordants. You have to use the glue for coating the stain to make it more visible. And the mordants here used is the tannic acid and the potassium alum. While you remember the mordant in the gram staining is the iodine solution. Paracyanilline Leifson method or basic Fuchsin method. So this is it for the staining, the Leifson staining, and these are not explained here, so I will go search it in a Google. Paracyanilline, Aniline. My gosh. So let's study what the gray method is. Fuchsin. What's wrong with my spellings? Fuchsin. Do they would know what we are talking about? Okay, I have to. Staining method, idiot. This is not working. I have to go for the Leifson method. What is the spelling? L-E-I, okay. Primary stain, basic Fuchsin. Mordant is tannic acid, sodium chloride, the final stain. Counter stain is methylene blue for better visualization. This is wet mount staining. Well, this is leaf staining. Wait, let's ask the ChatGPT. What is difference between Leifson staining versus what other did we study? Basic Fuchsin. Both laboratory techniques used for staining. Purpose is differentiation of bacteria based on their flagella. Used to stain is a general purpose staining technique to stain acid-fast organisms and some fungi spore-forming bacteria. Focus on staining flagella to study motility. Basically used for general bacterial staining. Leifson may use basic Fuchsin along with other chemicals. Basic Fuchsin primarily use basic Fuchsin dye. Why did they use it together? Leifson method or basic Fuchsin? Okay, whatever they say. So we are done with this, and maybe by the next day we will be studying electron microscope. Good luck, goodbye.